Yes, I think. Good morning, everyone. We're gonna start today's class. So we have here today a PhD candidate, Said and Elsafi. His seminar his seminar is Pathogenesis and the Treatment of Canine Prostate Cancer. Uh, Dr. Tom Lasso is his advisor. I'm let Dr. Lasso to have a brief introduction to Seth.
Thanks, Durong. Well, good morning, everybody. It's my pleasure to introduce Saeed Elshfei, who's a veterinary pathologist from Benha University in Egypt. And he's been with us 4 years and has done a tremendous job on pushing our prostate cancer project. So he got his veterinary degree and his Master of Science degree in Egypt. And he's going to get his PhD from The Ohio State University. And Saeed has the challenge today of, sharing almost 4 chapters of research with you. He's not going to try to show it all to you today, but show you an overview of 3 of his chapters and go into depth on a couple of them. So I hope you enjoy his presentation. He should be finishing his PhD this summer. Thank you.
Thank you. Do you hear me? Do you hear me, guys? So thanks, Dr. Russell, for this nice introduction. Good morning, everyone. I hope you're doing very well today. So today I'm going to present some data that I got so far in my PhD projects, kind of highlighting the chapters that I include in my PhD dissertation, and hopefully I can defend my PhD dissertation this coming summer. So before heading to each chapter or each project, I'd like first to give you a background slide about prostate cancer, how important is the prostate cancer. So as you know, prostate cancer is the most common malignant tumor among men in Western countries, and the second leading cause of cancer-related death after lung cancer among men. Almost 25,000 of new cases were diagnosed in 2015, and 30,000 will die as a result of this disease at a certain point. So in my PhD dissertation, I touched on many aspects of prostate cancer, starting from studying some signaling pathway in prostate cancer pathogenesis, like the first chapter. I studied the role of GRPR signaling in prostate cancer. Also, I studied, evaluated some drugs in treatment of prostate cancer metastasis as AR42. And the third chapter, we developed a novel prostate cancer cell lines that's similar to human prostate cancer in terms of bone metastasis. And we performed pathological studies on canine prostate cancer in the fourth project. So the first chapter, the topic of the first chapter is GRPR promotes EMT tumor growth and invasion of prostate cancer. And this chapter has been accepted for publication very recently under this topic. So what is GRPR? GRPR stands for gastrin-releasing peptide receptor. It belongs to G protein coupled receptor superfamily. Also, it's a subtype of Bombesin receptor. It's actually Bombesin receptor type 2. It's also 7 transmembrane receptor, which means it spans the cell membrane 7 times. And based on literature, we found interesting information from literature. So they found that GRPR is not only highly expressed in prostate cancer, in 90% of prostate cancer, but also expressed in many cancers, like 75% of pancreatic cancer, 90% of lung cancer, 50% of breast cancer, 100% of head and neck cancer, and 85% of brain cancer, especially glioblastoma. Also, they found that GRPR expression is very low to absent in normal prostate. Although that they found it's a very highly expressed in all stages of prostate cancer, starting from early stage of prostate cancer like PIN, which is prostate intraepithelial neoplasia, to advanced stage like invasive carcinoma and bone metastasis and lymph node metastasis. So we came up with a hypothesis based on this information that GRBR signaling is very important in prostate cancer metastasis. It enhances the proliferation, invasion, and metastasis of prostate cancer. And by activation of GRBR signaling, the epithelial phenotype of the tumor cells can be converted to mesenchymal phenotype of tumor cells. And this process called EMT stands for epithelial mesenchymal transition. And by acquiring the mesenchymal phenotype, the tumor cells are able to invade and undergo metastasis. And by blocking the signaling pathway, we can get the opposite with conversion mesenchymal phenotype to epithelial phenotype. And this process called mesenchymal epithelial transition, or MET. So we had some aims to test our hypothesis. So our in vitro aims were to determine the effect of GRBR signaling on tumor cell proliferation and migration, also to evaluate the expression level of EMT genes after activation, as well as blocking of GRBR signaling pathway. And for in vivo study, to determine the effect of Bombesin on tumor growth, invasion, and metastasis of prostate cancer in nude mice. To achieve our aims, we performed cell proliferation assay, wound healing assay, real-time PCR, and we did statistical analysis for our data. We used two agents, one agent to induce or activate GRBR signaling, as this was Bombesin, and the other agent was to block GRBR signaling, and this was RM26. This is a novel antagonist that has been developed in Dr. Tweedle lab, one of our collaborators. And we use two kinds of cells. We use ACE1 cells. So ACE1 cells is a novel canine prostate cancer cell line that was developed in our lab. And ACE1 human GRPR cells, it's ACE1 cells transfected with functional human GRP receptors for our study. So our results showed there was morphological difference between ACE1 and ACE1 human GRPR cells. So as you see on the left, ACE1 cells tend to be polygonal, adherent to each other, forming cobblestone pattern. While on the right, ACE1 human GRPR cells tend to be more spindle, non-adherent, and elongated cells growing out of the cluster radially. Also, by activation of GRPR signaling, we found that this enhanced the proliferation and the migration of ACE1 human GRPR cells. So as you see, the graph on the left, you can see how the enhancement of the cell number, we got increase in the cell number here after addition of Bombesin in ACE1 human GRPR cells when compared with non-treated cell. And for migration assay or wound healing assay, you can see there is a big difference between bottom panel and top panel. So top panel represent ACE1 cells non-treated, so no Bombesin, no receptors. And bottom panel represents the cells which have functional receptors and the agonist. So there was enhancement in the wound closure, as you see, by 24 hours. It was 0.1% of the wound and compared to 6% in the top panel by 24 hours. Also, Bombesin increased expression of mesenchymal markers. We tested 4 different mesenchymal markers in our study. We tested vimentin expression, twist expression, snail and slug, and all of them were increased after addition of Bombesin. So to make it easy for you guys, so the red bars represent ACE1 cells which don't have the receptors, and by adding the Bombesin, we shouldn't see any effect. While the green bars represent ACE1 human GRPR cells, which contain the functional receptors, and with addition of Bombesin, we should see effect in terms of increased expression in some cases or decrease. So vimentin expression was increased after addition of Bombesin, starting from 1 hour to 24 hours. And twist expression was increased also in these cells, in ACE1 human GRPR cells, starting from 1 hour to 24 hours. And the same thing happened to snail and slug, with marked increase in snail and slug expression after addition of Bombesin. And by using antagonists to block GRBR signaling, we were able to reverse the induction of EMT. We reversed the EMT to MET. So we can see the opposite happened here. So there was decrease in the expression level of mesenchymal markers like vimentin and snail, while there was upregulation of some epithelial markers like E-cadherin on the right. We performed a Vivo study to make sure that Bombesin can be considered as an agonist and induce the tumor growth. So we performed sub-Q injection and intratibial injection of nude mice with ACE1 human GRBR cells, and we divided the mice after injection equally to two groups. One group was receiving Bombesin intraproteinally daily, and then we performed bioluminescence imaging once weekly and histological studies and radiographic imaging by the end of the experiment. So our results showed that Bombesin increased the tumor growth and weight. So we can see the graph on the left, how Bombesin increased the tumor volume starting from 18 days and 21 days in the Bombesin-treated group versus control group. And on the right, we got heavier tumors by the end of the experiment in Bombesin-treated group versus a control group. For bioluminescence imaging, you can see there's a big difference between bottom panel, which represents the Bombesin-treated group. So at 14 days and 21 days, you can see there was increase in bioluminescence intensity in Bombesin-treated group versus control group. Also, there was morphological differences between the neoplastic cells in control group versus Bombesin-treated group. So you see on the left, the neoplastic cells tend to be rounded in the control group with large area of necrosis. And on the right, Bombesin-treated group, the neoplastic cells tend to be more spindle and with less necrosis. The spindle, this indicates like that the neoplastic cells underwent EMT. And also in Bombesin-treated group, we found that Bombesin increased the invasive power of the neoplastic cells, and this is indicated by infiltration of neoplastic cells among adipose tissue and among muscle. Also, Bombesin changes the phenotype of tumor metastasis. In control group, as you see here, and so here, so there was increase in radiodensity of the tibia, and this is reflected by increased osteoblastic proliferation with new bone formation. While in Bombesin-treated group, we found foci of radiolucency in the tibia with reduction of radiodensity, and this is reflected with osteoclastic resorption, as you see. Summary: Bombesin increased the tumor cell growth, enhanced migration in vitro. Also, Bombesin upregulated the EMT markers that were regulated epithelial markers, and the antagonism reversed the EMT to MET. And tumor derived from Bombesin-treated group grew faster and larger than non-treated group. Conclusion: GRBR signaling is important in prostate cancer progression, and it can be used as a useful target for imaging and treatment. Going to the chapter 2. So we evaluated the effect of histone deacetylase inhibitor, AR42, in prostate cancer metastasis. So AR42 is a novel histone deacetylase inhibitor that was developed in Dr. Shin Chen lab. He's a professor in College of Pharmacy. And AR42 was obtained from Arnold Therapeutics for performing our study. So background slide about histone deacetylase inhibitors. So histone deacetylase inhibitor stops tumor growth and induces differentiation and apoptosis in many cancers. Its mode of action either by reversing the deacetylation that occur in the tumor suppressor genes, in the promoter of tumor suppressor gene, or acetylation of non-histone proteins like heat shock proteins. Also, it suppresses angiogenesis by downregulation of some genes like VEGF and HIF-alpha. And AR42, as a member of histone deacetylase inhibitors, was found to be potent in many cancers like multiple myeloma, leukemia, and hepatocellular carcinoma. But the gap here, there have no studies done on the efficiency of AR42 in slowing down or prevention of skeletal metastasis induced in prostate cancer. So we came up with hypothesis that AR42 will shut down the expression level of AMT, analogous-resistant genes and metastasis genes due to epigenetic reprogramming, and this will result in lower progression and metastasis. So our hypothesis was based on once the tumor cells acquire mesenchymal phenotype, at this time it also acquire other properties like analogous resistance, can undergo invasion and metastasis, and can evade immune system, and can acquire also stem cell properties. And AR42 will block this transition to a mesenchymal phenotype, and thus reducing all these properties. So our objective were to assess morphology, cell viability, invasion, and metastasis of prostate cancer cell line, which was AS1, after treatment with AR42. Also evaluate the gene expression of AMT, analogous resistance, and metastasis genes in AS1 treated cells. Also investigate the therapeutic efficiency of AR42 in reduction or prevention of skeletal metastasis in not mice previously injected with AS1 cells. So we performed a cell proliferation assay, cell death detection assay using ELISA, also in situ cell death detection assay using immunofluorescence, and cell invasion migration assay and real-time PCR. So our results showed that AR42 inhibited the proliferation of AS1 cells in a dose-dependent manner. So the higher the concentration of AR42 reduced the lower viability you can see. So 1 micromole of AR42 induced more cell death or reduced the viable cells when compared with 0.1, for example. And IC50 was 0.4 micromole in our study. AR42 also induced apoptosis of AS1 cells. This is indicated by ELISA. So the graph on the left, the only graph that you can see, so represent the increase in enrichment factor. So enrichment factor means the amount of fragmented DNA released from apoptotic cells. So if you got a higher number in enrichment factor, this indication to the increase in apoptosis. So as you see, 0.9 to 1.1 micromole of AR42 induce more cell death or apoptosis in AS1 cells. And by application of certain equation to the numbers that you got in this graph, you can derive another graph talking about cell viability with the same effect. So you can see also 0.9 to 1.1 induced decreased the cell viability percentage. Also, we performed in situ cell death detection assay using immunofluorescence. So in control cells, you can see just a few apoptotic cells. So apoptotic cells usually appear brilliant green rather than the others, which is dark green. So at 0 point, we didn't see we saw few cells underwent apoptosis, but after 1 hour of AR42 treatment, we almost more than 1/3 of the cells underwent apoptosis. 6 hours, more 1/2 of the cells underwent apoptosis. And by 48 hours, you can see any viable cell. And in this experiment, we use higher concentration of AR42, which was 10 micromole. Also, AR42 suppressed AS1 cell motility, so you can see the graph on the left. So this represents the migratory cells or invasive cells. So this represents migratory cells. So the purple color represents AS1 cells. And during the assay, we watched already non-migrated or non-invasive cells. So what you see is just migratory cells and invasive cells here. And by treatment with AR42, we got reduction in both migratory cells and invasive cells. Also, by our result in real time showed that AR42 downregulated the mRNA level of some epithelial markers as E-cadherin. So this was greatly decreased after 24 hours. but there was no change in p-cadhenin level. Also, AR42 modulated the expression level of mesenchymal markers. So there was no change in vimentin, but twist expression was decreased starting from 6 hours to 24 hours. Snail and slug have the opposite happened in the snail and slug, so with marked increase in the snail and slug expression after AR42 treatment. And also ZIP1 was dramatically was increased, after 3 hours to 24 hours. Also, AR42 down-regulated the osteomimicry genes in ACE1 cells. So osteomimicry means the tumor cells resemble bone cells in terms of behavior, gene expression, secretion of some factors. So we found that AR42 down-regulated these characters in ACE1 cells. And this is reflected by decrease in expression level of osteomimicry genes, like osteoprotegrin was decreased after AR42 treatment. Also osteobontin, RANX2, and RANCO was greatly decreased after AR42 treatment. Also, AR42 downregulated the anoicus resistant genes, so there was marked reduction in BI3K expression and SARC and nuclear factor kappa B. And upregulated the positive regulator of anoicus, like P10 and PTK2. Also, downregulated some metastasis genes as MyoF. This was greatly decreased starting from 6 hours and 24 hours. There was trend for VEGF8 to be decreased, but it wasn't statistically significant, as you see. And also, we performed in Vivo Study to see the effect of AR42 in prevention of skeletal metastasis induced by ACE1 cells. So we performed intracardiac injection of in some of nude mice. It was 17 live animal, and we divided the groups. We divided the mice into 2 equal groups. One group was receiving AR42 orally daily at dose of 25 milligram per kilogram of mice, and we performed bioluminescence imaging, Faxitron imaging, histopathology, and immunohistochemistry for Ki67, caspase, and CD44. So our results show that AR42 decreased the number of metastases. So you can see the mice, we got 3 sites of where the signal of bioluminescence. So we got signal here on the head and 2 signal in the joint. Actually, this joint represents the distal femur and the proximal tibia. It wasn't the joint itself. And we excluded 1 mouse here due to unsuccessful injection. But after AR42 treatment, we got very weak signal in the joint, as you see here, very weak signal in the joint, and with no signal at all at the head. So there was reduction in the number of metastases. And this is reflected by the graph that AR42 treated mice showed reduction in bioluminescence by the end of experiment. Also, AR42 induced apoptosis of ACE1 cells in bone. So the picture on the left represents ACE1 cells, more spindle, adherent to each other, while on the right, in AR42 treated group, you can see ACE1 cells detached from each other. Some of them underwent apoptosis. Also, it changed the morphology. So ACE1s tend to be more spindle, but on the right, you can see AR42 treated group, the cells more rounded and build cytoplasm and build nucleus. Summary, AR42 decreased migration potential of ACE1 cells and induced anoicus and apoptosis of ACE1 cells. Down-regulated epithelial markers, twist, and anoicus resistant genes, reduced the tumor incidence and the number of the tumor in vivo, and diminished the osteomimicry of ACE1 cells by down-regulation of bone-related genes expression. Going to the 3rd chapter, development of a novel prostate cancer cell lines that's similar to human prostate cancer in terms of bone metastasis, and we named this cell line as LUMA. So background slide, we know that osteoblastic bone metastasis is very common in prostate cancer patients, and no animal model until this moment that can recapitulate all the stages of prostate cancer in human. And the level of RANXC2 in prostate cancer is positively correlated with the aggressiveness or Gleason score in human. Also, RANXC2 induced many genes responsible for angiogenesis, invasion, and metastasis in primary tumors. So the ultimate goal in our study is to develop a prostatic cancer cell line that can be used as a novel mean for understanding prostate cancer pathogenesis and also for testing new therapeutic agent. And after we got the cell line, our aim were to characterize the cell line in terms of gene expression, tumorigenicity, and identify the phenotype of bone metastasis. So we performed many assays to get LUMA identified. So establishment of the cell line itself, going to transduction of the cell line with luciferase for bioluminescence imaging, histology of, we performed some histological studies on the primary tumor and xenograft tumor, and also we performed real-time PCR for the gene expression, osteoblastic maturation assay, Calvaria co-culture, and Travis staining and alkaline phosphatase staining. So LUMA is derived from primary prostatic carcinoma coming from 13 years old castrated mixed breed dog. As you see, it's mostly multifocal, well-demarcated intraductal prostatic carcinoma with transitional differentiation, and some areas showed dysplasia and liquefacting necrosis. And tissue culture characteristics, it contains like two different populations. You can see the more spindle or angulated cells and larger cells in the tissue culture. And in terms of gene expression, LUMA has the highest expression level of osteomimicry genes. So LUMA has higher RANX2, osteobonin, rankle if compared with other prostate cancer cell lines that we have in our lab. Also has higher level of E-cadherin. This means it's more well differentiated than the others. Also higher level of MyoF. This means that has invasive power than other cancer cell lines, and higher level of CD44, which means also it has stem cell properties. So to see the effect of LUMA on osteoblast in vitro, so we used immature osteoblast cell line, which named as MEC 3T3 cell line. And then we incubated this cell line with the media previously incubated with LUMA. So our hypothesis in this experiment that LUMA will release some factors in the media, and by incubation this media with this cell line, we should see some, an effect. So what we saw maturation of this cell line after incubation. And also we performed the Calvaria experiment. So basically we extracted the Calvaria from 6 days old pup and removed the brain, make 2 holes in parietal bone, and incubated the Calvaria with LUMA cells plus BGGB, which is considered as bone media. And after 5 days, we took images using dissecting microscope, inverted microscope, and also Travis staining, alkaline phosphatase staining, and fluorescence. So without any staining, after 5 days, we got some growth on the border of the holes that we made. So you can see this is the border, roughly. This is the border that we made, and we got some growth here on this hole. And at this time, we have no idea what kind of cells are growing on the border of this hole. So we started to think about osteoblast. We performed alkaline phosphatase staining. As you see, most of these cells shown in bluish coloration represent osteoblast. Also, we perform trappist staining to make sure if there is osteoclast also in this population. So you can see just a few cells are osteoclast. And we perform a mixed alkaline phosphatase and trappist staining, and you can see the vast majority was osteoblast. Also, we performed immunofluorescence for alkaline phosphatase, and you can see how the growth of osteoblast, the brilliant green structure here, just growth of osteoblast on the border of the calvaria itself. Also, to see the behavior of Luma cells in vivo, we performed all routes of injection that can recapitulate the stages in human prostate cancer. So we performed sub-Q injection, intratubular injection, and intracardiac injection. And also at the end, we performed Faxtron or X-ray, histology, and IVIS imaging. So as you see, the graph on the left, the tumor growth, the tumor volume increased starting from 3 weeks to 4 weeks. And by doing histology for the xenografted tumors in sub-Q experiment, we found that the tumor behaves or similar to the tumors that we got in the dog in the primary tumor. And the same thing also here. So it's mostly prostatic carcinoma with transitional differentiation, but some tumors show papillary projection, as you see on the right. For IT experiment, Luma induced newly bone formation, and this is indicated that we mentioned in real-time PCR, Luma has the highest osteomimicry genes. So Luma can induce bone cells. So as you see on the left, this is Luma cells induced new woven bone formation, as you see here. And also here, you can see the Luma cells induce osteoblast proliferation and new bone formation. For intracardiac experiment, we found that bone and adrenal gland are most common sites of metastasis for Luma. And this is reflected, you can see the tumor mass in the adrenal gland here with necrosis and hemorrhage, and how the Luma cells distributed between bone trabecula in the bone. And we performed immunohistochemistry for RANX2, and we got higher expression of RANX2 in the cytoplasm of Luma adherent to bone trabecula here, and also in the xenograft sub-Q experiment. The same thing. The brown coloration represents RANX2 expression. Summary, Luma expressed higher level of RANX2, osteobonin, E-cadherin, rancol, MyoF, slug, and neuromedin B receptors. Also, Luma induced osteoblastic proliferation and bone growth in vitro and in vivo. Bones and adrenal gland are the most common sites of metastasis after intracardiac injection. Going to the fourth project, pathological studies on canine prostate cancer. I wish I have time to present the data of the fourth project, but I'm going to just highlight the study plans that we designed for this project. So the main aims were to propose new classification system of canine prostate cancer and set a scoring system that's similar to Gleason scoring system in human prostate cancer. Also characterize prostate intraepithelial neoplasia stemness in canine prostate cancer specimen. Also identify Bombesin receptors in this specimen. Also trying to find proliferative inflammatory atrophy, PIA, and differentiate between prostate cancers that arise from acinar versus dactylar versus urothelial cells. And this, I'd like to finish by acknowledgement my PhD committee, Dr. Russell, Dr. Dewell, Dr. Tribute, Dr. Shepsey. Also, I'd like to thank all my lab mates, especially Wessel. Also, I'd like to thank Washi, Pardis, Nicole, Eileen, and Lucas. Previous members, Jess, Ethan, James, and Rachel. And also, I'd like to thank Dr. Tweedle, Dr. Ding, Dr. Shin Chen Chen, and my Egyptian government for giving me this chance to be here today. And by this, I'd like to thank you so much and for attendance. And I'm very happy to receive any questions.
Questions?
Yes, Jim.
So I don't understand the new bone formation with the luma cell. Is that a characteristic of human prostate cancer metastases that they lose bone formation?
Yeah, so the question is osteoblastic bone metastasis is a common feature in human prostate cancer. So, yeah, according to literature, the most common feature of bone metastasis in human osteoblastic bone metastasis. And, but according to what we hear about prostate cancer in human, they say that most prostate cancer patients are suffering from fracture. And this can give indications that the vast majority should be osteoclastic bone metastasis, not osteoblastic. But the osteoblastic bone metastasis, usually the new bone formation is mostly new woven bone. Woven bone is susceptible for fracture than the normal bone. So although there is new bone formation inside the bone itself, but it's kind of weak bone. So although that also prostate cancer patient usually susceptible for fracture, not because osteoclastic resorption, but rather than formation of new bone formation, which very weak and susceptible for fracture.
More questions? In the first chapter, you show that the cell transfected with the receptor, the cell morphology, there's a big difference.
Yeah.
Do you have control show that cell transfected with a vector, for example? Now you can compare the differences in the cell.
This is a very good question. So we saw difference between ACE1 cells versus ACE1 human GRPR cells. And the question is if we have some cells transfected with a vector, not with a receptor, and shows a difference between them. We have, and we didn't see any differences between the cells transfected with vector versus the control ACE1 cells. But I don't have it here in my presentation.
Okay. Yes, Jim.
So is it not possible to work with gastrin-releasing peptide?
It's not possible. So, so the main, yeah.
It's possible to work with that. If that was, it seemed like that was the hypothesis in chapter 1.
Yes.
That the receptor was expressed.
Yeah.
And then you went to a more general inhibitor, which is fine. But is it impossible to obtain that peptide for these types of studies?
No, it's not so hard. So let me repeat the question, see if I understood your question. So ACE1 cells has very tiny amount of canine GRPR. So we modified our strategy because we don't have cell lines that express these receptors in vitro. Although it's GRPR is highly expressed in all primary tumors that we get from species in prostate cancer, sorry. But once we get the cell line from the primary tumor and try to culture it, for several passages, we're losing these receptors. So we put some receptors in the ACE1 cells to trying to perform our study and see the role of the GRPR receptors in prostate cancer. But it's not so hard. Actually, we cloned canine GRPR both in ACE1 cells, and this is our next study. And Bombesin represent GRPR analog to the GRP agonist. So GRP is a natural agonist of GRPR. And Bombesin is a synthetic peptide we can use just for activation of GRPR signaling. And so we have the agonist, we have the receptor, so it's not so hard to study GRPR signaling.
So my understanding, why not use human cell, human prostate cancer cell, human normal cell, then you treat with drug, then see difference. Why would you use a canine cell for in the first chapter? You have to transfect the human receptor. The question, how much homology between canine receptor and human receptor?
Yeah, so the homology between,
Repeat the question.
yeah, sure. So why I didn't use human prostate cancer cell line instead of canine prostate cancer cell line for study, for performing our study. So this is the first part of the question. So we performed our study on canine cancer cell lines because we have a lot of cancer cell lines in our lab, and we're interested in canine prostate cancer, trying to, so it's like you're studying canine prostate cancer to provide an information for human prostate cancer. So you're getting information for both, like canine and human. And why we didn't study human prostate cancer? Probably because we don't have, or to my knowledge, there is no prostate cancer cell lines that express GRPR extensively that we did in our cell line transfected with human GRP receptors. So getting cell line and then transfect with human GRP receptors may be challenging for us.
So you might want to add, so we developed a dog model for prostate cancer, which is used in the trial, put these cells in the dogs and quite well. So now we have, we actually use this one and GRPR cells in vivo to study human actors. So it's pretty exciting. We actually selected 100% of that's probably
Okay. So this is the first part. The second part, I can't recall
The homology between receptor,
yeah.
Between canine and human, if you drug treated, so what we inhibit, because you have two receptors in canine, you have canine receptor and human receptor, right?
Yes. So so we have very tiny amount of canine GRPR in S1 cells. And also the first answers the first part of question. So the homology between GRP receptors in human versus dogs, nearly identical. It's a 90% homology between them. And the second part of question, I can't recall it again. So the second part of question is by using S1 cells transfected with human GRP receptors, although it has very tiny amount of canine GRPR, but it's still negative because we didn't see any response in terms of after adding Bombesin to S1 cells, we saw no change in the gene expression. And I'm not sure if I answered your question or not.
Yes, you did.
Okay.
Any more questions? Okay, thank you.
Thank you.
Don't forget, fill up the evaluation online or whatever.